Okay. Good morning, everyone. I hope you you all are having a good day. This marks the end of our first week together, and I think there's only, what, 5 weeks? So like 20% done with the class. Well, you're 20% done with like listening to the class. You still got, all the work to do, but, getting close. So let's get started. I wanted to just first mention I have to just slightly update the syllabus. I updated on Blackboard and I put an announcement on Blackboard. But August 3 and 4, I will only have a recorded lecture. We won't have a Zoom virtual lecture. I have to do like I have sleep problems, so I have to do like an overnight sleep visit for these 2 days. So like I physically won't be able to come sit in class and stuff like that. So these 2 days are going to be just recorded lectures. They'll be posted on Blackboard like all the other lectures. But just as a heads up now, just as an FYI. All right, with that out of the way, let's get started. So we're going to talk about DNA today, or the genetic code. And so this is a sort of a foundational topic, like the previous 2 classes we've done. And really, this is really the most biology thus far we've had. And, the genetic code is sort of a really interesting set of things. It's something we've hypothesized to be a thing for 200 plus years of science. And it was really only within the past 50 or 60 years that we've really put a finger on what's going on with our genetics, how do our cells work, how do they carry information, and how do they transmit information to the next generation. And so, like, historically speaking, when people would talk about, heredity, they wouldn't quite know what heredity was. Like, what were the means of heredity? What were the means of transmitting traits to the next generation? And so the answer to this question is really just DNA, the genetic code, how we store our information. But ultimately, for DNA just to store information, that's great, but it doesn't really do anything for you as an individual. DNA is just simply the storage of your information. Everything that is you, your hair color, your height, the way you metabolize stuff, everything is just your DNA. But you have to physically take that DNA, that stored message, and essentially translate it into an active message, and that's in the form of RNA. And then finally, when you have that message, that RNA, you have to translate that into action. And that action inside your cells is what we call protein. And so we're going to work through this what's called the central dogma of biology this class. Talk about how DNA is organized. Talk about how DNA is essentially made into a message, and how that message is put into action inside your cell and inside the cells of every living thing on this planet. So DNA, I'm sure you guys have all probably heard of DNA. This little guy here on the right is Mr. DNA from the original Jurassic Park movie. So for those of you that have seen that one, he's the guy that says, bingo, dino DNA. But DNA stands for deoxyribonucleic acid. And it essentially codes everything you do. As I mentioned, your hair color, height, everything is encoded by your DNA. And even sort of weird things that we didn't quite know were encoded by DNA, such as behavior, as we'll find out later in the semester, are encoded in some fashion in your DNA. So your DNA really dictates who you are. DNA, as I mentioned, was just really only discovered within the past 60 or so years, and it's credited, the people that are credited with its discovery are James Watson and Francis Crick. They built heavily upon sort of the work of others, but in the early 1950s, they did receive the Nobel Prize for the elucidation of the structure of DNA. We sort of have this little iconic image of Watson and Crick here, sort of pointing out in their model sort of what the structure of DNA is. Because remember, prior to the 1950s, we had no idea what the structure of DNA was. And this was a huge advancement, arguably one of the most important advancements of the past 100 years, scientifically speaking. But as I mentioned, they did build upon the work of others, and one of the most important people was actually a woman named Rosalind Franklin. And unfortunately, Rosalind Franklin was doing her work at a time where many women scientists were sort of just kind of thrown to the back. Their work wasn't really taken seriously. and their work was sort of overtaken by, unfortunately, men. Sexism was pretty rampant in the pre-1980s scientific world. It's not rampant still, but sexism still does exist in science, unfortunately. But in particular, Rosalind Franklin made this absolutely monstrous discovery by looking at X-rays of DNA, and she's the one that really came up with this sort of X-like structure, which then pointed to the idea that DNA is a twisting spiral. Because the only way you can really sort of figure out that DNA could take the shape under an X-ray is if it was spiraling around. So major props to Rosalind Franklin. She's a very fascinating figure in science. So if you have a free minute, I would recommend reading up about Rosalind Franklin and how awesome she was. But DNA is stored as, it sort of has multiple levels of organization. The highest level of organization is what we call the genome, and many of you probably heard of this term genome, like you might get your genome sequence, or maybe you heard of the Human Genome Project. And when we say genome, it's simply the collection of all DNA inside of any given cell. That's it. So all the DNA inside one of your cells, that's what we call your genome. Your genome, like pretty much everything else on the planet, is organized into what we call chromosomes. And chromosomes are just one of, essentially just smaller subsections of your genome. It's really it. Nothing really special to sort of defining what a chromosome is, just a small section of your genome. Typically, it's going to be one linear section of your genome that's distinct. Its own sort of like distinct part from the rest of your chromosomes in your DNA. I'm sorry, in your genome. And then finally, the smallest unit of your chromosomes is going to be genes, which are specific DNA sequences. They're typically a few thousand DNA bases long, and they contain the information necessary to make proteins. And remember, those proteins are the mode of action inside your cell. And so DNA, as I've mentioned before, it forms the double helix. It's essentially like if you took a ladder and you twisted it up. And it has repeatable overall structure. It has what we call these tight turns, and then it has what these sort of wide turns. And this sort of distance here between, say, this piece of DNA here, and this piece of DNA here is conserved essentially throughout the entire length of your chromosome, as well as the entire length of any organism's chromosome. So this structure DNA takes, the turns, how much it turns, the tightness of the turns, is actually remarkably consistent across all life forms on the planet. DNA itself is composed of 4 bases, and you'll see these bases down here at the bottom: adenine, thymine, cytosine, and guanine. So everything in your DNA is coded by those 4 things, repeating in some fashion. And I think that's sort of a remarkable thing to think about, right? Your DNA, everything that is about you, your incredibly complex biology of 38 trillion cells, is all controlled by changing the order of 4 DNA bases. I think that is absolutely crazy to think about, right? Think about how complex life is. Think about how complex we are. The thinking we can do, mathematics, singing, everything is controlled simply by the order of 4 bases. And I think that's absolutely crazy. Maybe it doesn't blow your mind, but it certainly blows my mind. That being said, all life as we know it does use these 4 bases. And what this suggests to us is actually that this is something that evolved very, very early on in the history of life on Earth. And it's something that was so successful that it never deviated at all. There's no life on this planet as we know it. Maybe there's life on other planets. But all life as we know it uses these 4 bases and keeps the same conserved structure of DNA. And it's really, really remarkable that it does this. There are some sort of rules to the way DNA works. They're called binding rules, and DNA is actually held together by hydrogen bonds. So remember those bonds that occur between partially positive and partially negatively charged things. So you'll see that this negatively charged oxygen here is binding to that partially positive charged hydrogen here, and same here. Same here, we have the hydrogen bonding to nitrogen, and oxygen to hydrogen, nitrogen to hydrogen, oxygen to hydrogen. So all your DNA, that ladder is held together by hydrogen bonds. So this is why hydrogen bonds are really important, just at your cellular level. But that being said, there is ways that these bases have to pair. So our base adenine always pairs with thymine, and our base guanine always pairs with cytosine. There is no other way for these bases to come together. Adenine can't bind with adenine. Thymine can't bind with guanine. There's no other combinations that work here. Adenine can only bind with thymine, and cytosine can only bind with guanine. That's the way it works. That being said, there can be sort of weird things that happen, but typically speaking, your cells will essentially fix these errors because they just don't, they sort of harm the overall structure of the DNA and can cause some pretty serious side effects. But again, there's some pretty conserved base pairing here, A with T, G with C. And again, one of the sort of the things about these is they're actually relatively conserved in their overall chemical structure. Remember, DNA is just a bunch of chemicals. Remember, life is all about chemistry. It's all about bonding. And your DNA is no different. And so DNA bases have a conserved overall structure. So they have what's called a sugar phosphate backbone. They have a sugar here. You'll notice this looks like glucose that I've been showing you from our chemistry slides. And that glucose is attached to what's called a phosphate group, or just phosphorus. And you'll recognize this actually is what our energy-carrying molecule, ATP, had 3 of. Right? So this is a phosphate group because it's a phosphorus attached to 4 oxygens. So this is the sugar phosphate backbone. Every DNA molecule has this exact same sugar phosphate backbone. And connected to that backbone, it has what we call a nitrogenous base. And this nitrogenous base is what gives the DNA molecules, the DNA bases, their unique properties. So slight variations in this base give you your adenine or your guanine or your thymine. I'd like to also point out is that this sugar backbone here is what really makes up sort of the outside proportion of this ladder. Sort of, the rails, I guess that would be. I think that's what they're called. The rails are these sort of the outside portions of the ladder, and the nitrogenous bases make up the sort of the rungs of the ladder. And so sort of moving on, thinking about DNA as a whole. So we're going to start with bacteria. So you all have heard of bacteria at some point, but bacteria also have a cousin. Well, we also have the same cousin because it's related to us as well. So bacteria, as well as archaea, they're microscopic single-cellular organisms. Bacteria you guys all know and love because they make you sick. So you think of something like food poisoning or something like pneumonia, that's bacteria. But their cousins, archaea, they're single-cell organisms, and we're going to talk about these guys in a couple classes. But these 2 organisms have a single circular chromosome, and you can see that here. Excuse me. And our microorganisms can also have what we call accessory chromosomes called plasmids, and these are very, very small circular rings of DNA as well. And plasmids, they carry, I say cool stuff because, like I said, like I told you guys, I'm a microbiologist, so I think microbes are cool, but plasmids are known to carry things like antibiotic resistance genes, genes that make you sick. So plasmids carry all sorts of nasty stuff, but they are an important part of the bacterial genome. So remember, a genome is all the DNA in your cell, and chromosomes are sort of individual slices of that DNA. And we don't really consider plasmids to be chromosomes. They're too small overall, just as a note. Now, eukaryotic DNA, and so sort of I should take a step back. Eukaryotes, which is sort of encompasses everything that's not a bacteria or an archaea. We are eukaryotes. Cats are eukaryotes. Plants are eukaryotes. Anything you could think of that's big is a eukaryote. And we'll go through sort of the classifications of life later on in the semester, but just as a note, humans are what we call eukaryotes. But humans, like all animals and plants, have multiple chromosomes. Now, in particular, human DNA, our large genome, which consists of about 3 billion DNA bases, so 3 billion A, T, Gs, and Cs, our genome is split into 23 pairs of chromosomes. So grand total, each of us has 46 chromosomes inside each of our cells. As I mentioned, your chromosomes are paired. So you have 2 copies of each chromosome. So for instance, you have 2 copies of chromosome 1, 2 copies of chromosome 2, and so on and so forth. Your chromosomes hold all the directions to encode all your proteins in your genes. And remember, genes are what makes proteins, and proteins are what are the actions inside the cell. Your paired chromosomes, they're not just called paired chromosomes, they're also called homologous chromosomes. That's just a fancy term. Homo meaning same, so same exact chromosomes. So every chromosome in your body, you have an identical or nearly identical copy of your chromosome. Other eukaryotes have varying amounts of chromosomes, so this sort of overall picture of a picture of chromosomes in an individual is actually what we call a karyotype. And we have a fruit fly here, and we have corn. And what you'll notice, corn has 10 chromosomes, and then some weird chromosomes that are involved with determining sex and making sex cells and things like that. But what you notice, it has 2 copies of chromosome 1, 2 copies of chromosome 2, and so on and so forth. That's in contrast to our fruit flies, which have, you guys all know fruit flies. They're the little buggers that you'll see flying around. Actually, you'll see them flying around McCormick Hall because they use them in the genetics lab there. But you also see them around like dead, rotting fruit in your trash cans. They're little teeny flies. They're kind of a nuisance. But our fruit flies, they only have five regular chromosomes and then 1 pair of what we call sex chromosomes. And you'll notice is that they are paired like our chromosomes. So different organisms can have different numbers of chromosomes overall. And because we have essentially 2 copies of each chromosome, I'm just going to hop back a slide, we have 2 copies of every gene present in our genome. So if I have a gene that encodes for my hair color, even though my hair is kind of going away, I have 2 copies of that gene. And 2 copies of any given gene are what we call alleles. And so all plants and animals have 2 alleles or 2 copies of each gene inside their genome. And each copy of the gene is carried on one of those pairs of chromosomes. And so bacteria, I will just mention at this point, bacteria typically only have 1 copy of each gene. Bacteria will sometimes have multiple copies of genes, but typically speaking, each bacteria only has 1 copy of every gene in its genome. But plants and animals, we're special. We have multiple chromosomes. We have paired chromosomes. Thus, for every gene we have 2 alleles or 2 copies of that gene. And one of the sort of important things about alleles is they can actually lead to novel traits. So for instance, in this case, where we're looking at flower color of a plant, the flower color of this plant here is simply determined by the proteins present inside the flowers. And we're going to talk about flower color later in the semester when we talk about genetics. But I think this is a good foreshadow of it. But what you notice is that we have different copies of the gene that make this plant's flower color, and they're encoded on different alleles. So they have slightly different DNA sequences leading to very different flower colors. And sort of as a last note about alleles can be absolutely identical, like the exact same string of DNA bases, or they can have slight variations or major variations. And major or slight variations are what give rise to these sort of differences in colors that we're seeing here in these these flower petals. And so just to bring it back to the sort of overall structure, genes have multiple alleles, and these 2 copies of alleles can potentially lead to different proteins. And your DNA is organized into chromosomes. And that sort of overall picture of your DNA is what we call a genome. And genomes are sort of an interesting field. You might not think it's an interesting thing, but I like these sorts of things, so I think it's interesting. Genomes vary in size. And sort of a remarkable thing is you can look at the number of DNA bases here. So this is a log scale. So we have 10 to the 5 here and 10 to the 12 DNA bases. So it ranges from 10,000 bases all the way up to, 12 billion bases, if I'm doing my math correctly here. And we have sort of different types of organisms. So we have bacteria, fungi, protists, plants, insects, mollusks, and so on and so forth, all the way down here with mammals at the bottom. And this bar sort of spans the genome size range that you typically see in these organisms. And so what you'll notice right off the bat is our smallest genomes are actually our bacteria. If I put viruses on here, viruses are down here. They're much smaller. They're only 10,000 bases or so. But what you notice is that our bacteria have the smallest genomes, and our plants actually have some of the largest ones. as well as protists, which are single cellular organisms. Fish also have relatively large genomes. But what you'll notice here is that humans are kind of in the middle, right? We're not huge, we're not small, we're sort of kind of in the middle. And there's many, 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 many more organisms on this planet that have much larger genomes than us. Typically every year when I do this in person, I ask everyone to sort of raise their hand and be like, hey, what do you guys think about this? Do you think this is weird? Did you expect humans to have the biggest genomes, right? Because theoretically speaking, we're the most complex, right? We're the only people that build buildings, right? We're the only people that think the way we do, but we have sort of a middle of the pack genomes. There are tons of fish out there and plants that have bigger genomes than us. Like for instance, like if you go to the store and buy a can of beans, that bean has a bigger genome than you, right? And so I always like to ask people, like, what do you think? Did you expect humans to have the biggest genome? Because theoretically we're the most complex because we have we have the capacity to build and think. Or did you expect us to have sort of smaller ones or sort of in the middle? And people will sort of have this range of things like, wow, I was expecting us to be the biggest. And, I say to that, like, well, you're not alone. When we first sequenced the human genome, we expected it to be massive. We expected to have the biggest genome on the planet because we were so complex. But as a matter of fact, we don't have a very big genome at all. And one of the important things to note here is that genome size does not correlate with complexity of the organism at all. You can have the biggest genome that we know of, which is a type of plant, and you are still not as complex as, say, a human. So genome size and complexity don't scale together. And so I find this graph to be fascinating. Maybe you don't find it to be fascinating, but it is a sort of nice look to see how genome size does vary, not only within the animal and plant kingdom, but also relative to, say, bacteria and something like a yeast, which we use to make bread or beer. And so one of the things that can artificially change the size, well, not artificially, that can change the size of your genome is this idea of coding versus non-coding DNA. And the idea being is you can have coding DNA, which directly makes a protein, or you can have non-coding DNA, which actually doesn't contribute to making the protein. And we'll get into that, what that looks like in a second. But what you'll notice is we have percentages of coding DNA found in various organisms. And you'll notice is that humans, only about 2% of our genome actually encodes for protein. The rest of the genome is what scientists have historically called junk DNA. DNA that doesn't actually physically make proteins. So 98% of your genome actually doesn't encode a protein. And remember, proteins are doing all the action in your cells. That's in contrast to E. coli, which is a bacteria. This is the fun guy that causes food poisoning, or I should say one of the guys that causes food poisoning. And E. coli's genome is 90% coding, meaning that 90% of all the DNA in E. coli cells encodes for some protein or another. And the reason humans and fruit flies and worms and plants all have low percentages of coding DNA is actually just the way our genes are set up. And so we can actually look at a gene inside of a human, and we have different colors here. So we have this dark blue, which is what we call an exon, and this light blue, which is what we call an intron. And that intron is what we call an intervening sequence. This is a piece of DNA that doesn't actually encode for the protein. So this little piece here is what we would call a gene. This encodes for one protein. And this DNA is ultimately transcribed to RNA, which we'll get into in a second. And that RNA actually has to physically chop out these non-coding genes, these introns. And so what you'll notice is that by the time we have this final product of RNA, we get rid of most of the DNA in this. So this is what I mean by most of your genome doesn't actually code anything. We have to cut out a lot of garbage before we can actually make a protein from your DNA. Just as a note. So that's DNA. We'll go back to the central dogma of biology again. DNA, the storage molecule of every living thing on the planet, has to be transcribed into a message. And this message is RNA. So let's talk about RNA now. So let's do a sort of a broad overview here. And the analogy we're going to take, trying to sort of make this analogy of how we go from the DNA, the sort of the storage of information, to the actual physical action, is we're going to use an analogy of cookies, of making cookies. So we're going to pretend that your grandmother is still alive and she's making you cookies. And so she has her cookbook of her cookies, and she wants to make her cookies. So she's going to take her cookies in her cookbook. She's going to copy the cookie to an index card. She's going to take that index card with the recipe, combine all those ingredients together, and then she's eventually going to bake them into chocolate chip cookies. So this is the sort of the workflow that grandma would do to make cookies, or anyone that would do to make cookies. And we can use this as an analogy for how we physically make things inside of our cell. And so we have our cookbook here, which is our DNA. And remember that DNA has genes on it, and those genes encode for proteins. Right? Basically everything in your cell is just trying to make proteins in some way or form. What we're going to do is, in a process of what's called transcription, or copying the index card, copying the recipe onto an index card, is we're going to take that DNA, we're going to bust it open, and we're going to make a copy of that DNA. This is what transcription is called. And that copy of DNA is what we call messenger RNA, or mRNA. Once we have that message, we're going to take that message and we're going to export it we're going to move it around our cell, and we'll get into the sort of the structure of the cells in a later lecture, but it's important to understand DNA before we really get into anything else. We're going to take that index card, or our mRNA, we're going to move it around our cell, and then what we're going to do, we're going to do a process called translation, where we're going to take that mRNA, that message, and we're going to read that message and combine a bunch of different ingredients together to make a protein. And so the analogy for our cookies, we're going to by taking that index card with the recipe, we're combining the milk, the sugar, and the butter, and all that fun stuff. We're combining that all together so that we can eventually bake those cookies and have our delicious cookies. And so that's the sort of the flow we're doing. DNA is the message. We transcribe that DNA. DNA is the storage information. We transcribe that DNA into a message called mRNA, and then we essentially move that mRNA inside of our cells. We perform a process called translation, where we take that mRNA, we read the message, and we make a protein, which is ultimately the sort of the game plan here. We take the message in the DNA, and we try to make a protein from it. So the process of reading our DNA and making that message is, again, called transcription, where we're reading the DNA to make messenger RNA. Now, RNA stands for ribonucleic acid. It's similar to DNA, right? DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. Ultimately, the RNA is going to be a direct complement of the DNA. The idea being is we need to use our DNA for stuff, but we don't want to damage our DNA. We don't want to get rid of our DNA. We want to keep our DNA intact, because without our DNA, our cells are completely lost. So to preserve our DNA, we make copies of it, right? So if you had like a textbook, right, and you wanted to take notes on it, but you didn't want to ruin your textbook, well, you could just photocopy your textbook and then take notes on the photocopy of your textbook, right? That's the idea here. We're trying to keep our DNA intact. Thus, we make a copy of our DNA, and that copy of our DNA is our messenger RNA. So RNA is actually almost identical to DNA. It uses the same sort of A, T, G, and C combination of bases. That being said, it does replace the thymine of DNA with what we call uracil, which is denoted with a U here. The physical copying of DNA into RNA is actually performed by an enzyme that we call polymerase. You don't need to remember that, but I just wanted to put that out there. You might hear that in some way when people talk about medicines or something like that. And again, RNA is a direct complement of DNA. So for instance, if I have a DNA sequence that is ATCG, RNA is the complement of that, meaning that RNA has to physically be able to bind to DNA. So thus, RNA would have the exact opposite of DNA, right? So remember our complementary base pairing rules. A has to pair with T, but RNA doesn't have T, it has U. So A would pair with U. T has to pair with A, so the RNA has to have an A. C has to pair with G, so the RNA has to have a G. And then finally, G has to pair with C, so RNA has to have a C. So what you notice is that that DNA, that RNA is a direct complement, meaning it can bind physically with the DNA. So you're basically taking the message and making a copy of it, almost like upside down. You could sort of thinking about that. Well, that's it. So that's what we're doing. We're taking the DNA, we're reading it with an enzyme called polymerase to make essentially a complement of that DNA. And that complement of that DNA is our messenger RNA. I just want to make a note about how messenger RNA is processed. So when a bacteria makes a messenger RNA, it uses that messenger RNA to directly make protein. It doesn't need to edit it at all, because the bacteria don't have those intervening sequences, those introns. Eukaryotes, us, and plants, and anything that's not a bacteria, remember, has to chop out those introns, because those introns are non-coding DNA. So just as a note, transcription in eukaryotes has to make what's called a pre-mRNA, and then chop that mRNA up to make a full mRNA, just as a note. Not that important, but figured I'd just make mention of it, because there is a pretty distinct dichotomy between our microorganisms and our humans. And so this is sort of what we've done. So the RNA polymerase has to recognize and bind to the DNA. And remember, RNA polymerase is just an enzyme. This enzyme reads the DNA, and as it reads the DNA, it makes an mRNA copy. And that's it. That's really all that's happening during transcription. It's making that complementary copy of DNA that we call messenger RNA. What you will notice is that as this polymerase is reading the DNA, it does have to break the DNA open. It has to sort of unwind the double helix, just as a note. So that's transcription, making a physical copy of your DNA. You're making that message. But now that we have the message, we need to make it into an action. And that action is essentially done by a process called translation, where we convert our RNA into a protein. And proteins, again, basically do everything inside your cell. Any chemical reaction that happens inside your cell is done by a protein. And remember, the purpose of life is basically just to do a bunch of chemical reactions, right? If you're going to do photosynthesis, you have to perform translation because you need proteins. If you're going to do respiration, you need protein. And so if you're going to be a cool you're going to be a cool fish and glow green, well, you need to make a protein to glow green. So proteins are the action inside your cell. And as we'll learn later in this semester, proteins are also structural components of your cell. But thinking of proteins as primarily as a way to do stuff inside your cell is a sort of a good way to wrap your head around it. And proteins, again, action in the cell, are made up of what we call amino acids. And you might have heard amino acids before. I see like supplements I've seen supplements in like Target and Walmart and stuff like that, that sell amino acids. They don't do anything, but they're there. And amino acids are these building blocks of proteins. And so there are 20 distinct amino acids, and these 20 amino acids make every single protein on the planet. They make all the proteins in you, me, blue whales, bacteria, yeast, everything. So everything on the planet uses the same genetic code, uses the same and uses the same 20 amino acids to do everything. And this suggests, again, like everything using A, T, G, and C, is that these amino acids evolved very early on in life, and where they're sort of propagated through time. And the idea being is if we're going to make a protein, we link together some order of these 20 amino acids together to make a protein. I froze. Oh, there we go, I'm back. And so amino acids, again, I mentioned there are 20 of them, and this is what they look like. They're just a bunch of carbon linked together. That's all they are. And these amino acids have very different properties. So some amino acids can be positively charged or negatively charged. Some can be very sort of large and what we would call hydrophobic, meaning they fear water. Others can be hydrophilic, meaning they love water. And there's all sorts of different properties these amino acids take. But what you will notice is that all these amino acids have almost the exact same structure. So every amino acid has this backbone where they have what's called the carboxyl group, which is a C bound to 2 O's and an H, so COOH. That's then bound to what we call a central carbon. And this central carbon is then bound to a single hydrogen atom. And then it's bound to what we call an amino group, which is just an N bound to 2 H's. Every amino acid has this exact same backbone. And just as a note, these are all covalent bonds, so again, equal sharing of electrons. What differs between each of these amino acids is this side chain, or this, what we call this R group. And this R group, what you'll notice here, varies pretty substantially. And so R groups can be as sort of complex as this, or as simple as just one carbon hanging off. And so very subtle variations cause these amino acids to have very different properties. But the properties of these amino acids are important for the properties of the protein you're trying to make. And so every protein has different amino acid sequences. So for instance, we'll look at a very important human protein. Everyone makes this. This is insulin. A gentleman named Fred Sanger in 1979 won the Nobel Prize for figuring out this amino acid sequence. And figuring out this amino acid sequence actually has transformed the way diabetics live their life, because by knowing the sequence here, we can make it, which means that diabetics should get more stable and cleaner insulin. But what you will notice is that human insulin, it's a double chain protein. Insulin is just a protein. And what you'll notice is that it has two chains, chain A and chain B, and it's not very big, right? The chain A is 21 amino acids long, and chain B is only 30 amino acids long. It's very short, but it's a very important protein. But what you'll notice is that they have different sequences of amino acids. So we have glycine and isoleucine and valine, and it's sort of just a different string of amino acids. The string of amino acids gives it its distinct property. Because what's going to happen with the string of amino acids is the cell is going to fold it. It's going to change its shape. And remember, structure and the shape of things dictates how they're going to function. So if you have a shape that looks one way versus another way, they're going to behave very, very differently. And that's going to be important for how these proteins function inside their cell. But again, all that difference between every protein on the planet is how you string together these 20 amino acids. And insulin is a small protein. Again, it's only got 53 amino acids. There are some proteins that have thousands of amino acids strung together. But insulin's small. It's easy to understand. But again, all that makes insulin what it is how you take those amino acids. Now, the question you should be thinking to yourself is, how do I get those amino acids? How do I know what amino acids to string together to make a protein? Well, each amino acid itself is coded by 3 RNA bases. These 3 RNA bases are what we call codons. So for instance, if we're going to make amino acids such as phenylalanine, so PHE here, we need 3 RNA bases, either UUU or UUC. That's how your cells know to make phenylalanine. Just by the presence of UUU in your mRNA sequence, those to make amino acid A versus amino acid B. And remember, that string, how you string together those amino acids directly tells you what amino acid to make and what amino acid to add into your chain of growing amino acids. And remember, all a protein is just a string of amino acids together. And so just to sort of tie this back to transcription, right? We started with our information stored in our DNA. We transcribe that into messenger RNA, and then we have to take that messenger RNA and then translate it into a protein. So that's the workflow, right? DNA, RNA, protein. And so remember, RNA is a reverse complement of DNA. However, the protein sequence that you ultimately make is going to be complementary to the RNA sequence. So the idea being is, if I have a DNA sequence that is ATCG, remember the RNA sequence that pairs with it is UAGC. The paired protein sequence, which then makes amino acids, would then be AUCG. So what you'll notice is that in our proteins, we also have that sort of, we've gotten rid of thymine. But what you'll notice is that our protein sequence here reflects exactly what our DNA sequence is. So the idea being is we took a message, we copied it, we flipped it over, and then we copied that message again during translation to be right side up. So now we're reading the message perfectly the way it's supposed to be read when we're making that protein. Kind of weird thing to think about, but you're taking the message, you're making a weird copy of it, and then you're reading that weird copy to make it right side up again. That's sort of the flow of information. I know it's kind of weird, but that's just the way our cells do it. And so the way we sort of do translation is by using a special type of RNA, which is called tRNA or transfer RNA. And now transfer RNA, it's a special type. It's something that sort of free floats in all of our cells, including bacteria. And tRNA is interesting. It has this T-like structure. And it's sort of interesting. It has multiple loops. But what you'll notice is it has what's called the anticodon loop. And that should ring a bell in your brain. So if we go back a few slides, I called a three RNA base is a codon, right? So UUU is a codon. 3 bases of an RNA is a codon. tRNA has the matching anticodon. And you'll notice that if I have my mRNA here, I have this codon here, it's GAG. Well, the complement to that would be CUC on my tRNA. Now you're thinking, okay, how does that, okay, all right, I get the tRNAs there, but how do I make my amino acids? Well, what you'll notice is that each anticodon, which again complements to a codon, has what we call an amino acid attachment site at the very top. And this amino acid attachment site is where a specific amino acid will bind to. And remember, each anticodon or each codon has its own specific amino acid associated with it. So the idea is we can use this tRNA in a special protein called a ribosome to string together a bunch of amino acids. So the idea being is we use these ribosomes, which kind of look like hamburgers, is we essentially feed in our mRNA into these ribosomes, into the middle of the hamburgers. We recruit tRNA that matches the codons on our mRNA. And remember, these tRNA have amino acids tacked on to the back end of them. So the way this works is again, we have our hamburger ribosome. We feed a messenger RNA into here. And remember that a messenger RNA has specific RNA codons on it. Remember those triplets that match with different anticodon triplets on our tRNA. So what happens is as we feed this messenger RNA through, it happens really, really fast, like thousands of bases every minute. As we feed it through, the ribosome, what it does is it recruits tRNA into the ribosome, matches them to the anticodon to the codon, and what it does is it takes the bound amino acid up here, attaches it to the one before it, and strings together a bunch of amino acids, thus creating the protein. And it does this sort of it doesn't read it right to left, it reads it left to right. I'm sorry, it reads it not left to right like we read a book, but it reads it opposite. It reads it like you would read like say the Torah, like you would read my goodness. Like you would say like you would read Arabic. You read it from right to left. And so the idea being is that the ribosome recognizes what we call a start sequence in the mRNA. And then once it recognizes that sequence, it then starts building the protein chain, which again are just these amino acids. Eventually it recognizes a stop sequence in the mRNA, so it knows to stop adding amino acids. And that's the idea. We have this ribosome that's reading the messenger RNA sequences, recruiting tRNA, which have these amino acids tacked onto it. And as it reads them, it starts building a protein chain. It starts building a chain of amino acids. And once it gets that chain of amino acids, it reaches the stop codon, it stops making the protein. And that's how you make a protein. You read the mRNA sequence, you string together a bunch of amino acid sequences based upon that mRNA sequence. And once you reach the stop sequence, you're done making your protein. And once the protein's done, it can then do whatever you need it to do inside of a cell. So it's a pretty nice flow of information inside your cell. You take that DNA, which is long-term storage inside your cell, you perform transcription. You make a copy of your DNA, which is RNA or messenger RNA. Once you make a copy of that DNA, you have your transcript or your RNA, and then you use that RNA inside your ribosomes in a process called translation to string together a bunch of amino acids to make what's called a polypeptide or a protein. And once you make that protein, then you can do whatever you need to do inside your cell. So that's the flow of information here. DNA, RNA, protein. That's how you do everything. And I'm less concerned with you remembering, you know, the very specifics here, but sure, there's so the difference between what somebody asked the question, what's the difference between RNA and mRNA? Well, they're essentially the same, right? So there are RNA is sort of a broad classification of things. So we have tRNA and we have mRNA, and those are both RNAs. And there's also a third type of RNA I didn't make mention of, but it's here. It's called ribosomal RNA. It's a structural component of your ribosomes. You don't need to remember it at all. Just as a note, there are 3 types of RNA. mRNA is a very specific type of RNA that acts as that message. It's mRNA, so messenger RNA. And that's what we're making here. I know this doesn't say mRNA or mRNA here, but that's what we're making. So mRNA is just a specific type of mRNA, a specific type of RNA, and there are 3 distinct types of RNA. That clear up the confusion there? I know it says RNA here. I should edit this. I like this figure because it's nice and simple, and it's slightly detailed, but I should probably edit that to sort of reflect that. Oops, my bad. But that's the sort of the flow of information. That's the sort of the key that I want you to think. This is how we go from your DNA, from your genome, to actual physical action. That flow, big picture stuff. And so sort of just to wrap up this this talk about DNA and how things work, I just want to sort of briefly talk about chromosomes one more time. And so we have our bacteria and we have our eukaryotes. And so remember, our bacteria just have one single chromosome. They're very simple, they're circular, they're relatively small. That's in contrast to eukaryotic cells, which have multiple chromosomes, they have much larger genomes. And one of the things that I just wanted to make note about the way chromosomes work is our chromosomes are actually what we call supercoiled around proteins called histones. And so what the idea being is you have your DNA, that double helix, you wrap it super duper tight around these proteins called histones, which kind of makes them look like Oreos. And as you supercoil, you sort of wrap it super duper tight around these histones, these proteins. That's how chromosomes condense and come together to make that sort of classical X shape that chromosomes take. So that's the structure of chromosomes inside your cell. That's in contrast to bacteria, which don't do any of this stuff. They don't wrap their DNA around histones. They just sort of have it like spilled out in the cell. In addition, I wanted to bring up a cool feature of your chromosomes, and it's a really fascinating area of research. And if you have about 15 minutes or so, I'd recommend watching this video. The woman whose name escapes me right now, she's an expert on these things called telomeres. And it's a really fascinating YouTube watch. So if you have a minute, well, if you have 15 minutes, I'd recommend watching this video. But at the end of every one of your chromosomes, you have these caps, and these caps are called telomeres. And one of the sort of interesting things we've noticed about telomeres is as you get older, as your cells grow and divide, they get shorter. So when you're born, your telomeres are long. They're nice, effective caps on the end of your chromosomes. But by the time you're middle age, they've shrunken dramatically. And by the time you're in your 70s and 80s, they're almost non-existent. So they get shorter as you age. And so it's one of the interesting things is that they're nice and long. They're caps on the end of your chromosomes. They're designed to protect your chromosomes. They don't code any proteins. But as your cells grow, as they divide, you start to lose little pieces of them. Eventually, if you lose the entire cap, you start to damage your own DNA. And it's what is sort of interesting is that as you get older, you start to damage your DNA. And as you start to damage your DNA, you start to show signs of aging, start to show signs of disease. So shortened telomeres are associated with aging and they're associated with diseases. And so there's people like the woman in this video that are actually researching ways to actually physically repair these telomeres so that people will stop suffering from certain diseases and potentially to halt the aging process. It's pretty cool. So that's why I think you should check out this YouTube video. Like I said, it's like 15 minutes. It's a really fascinating topic. But there is a set of syndromes associated with shortened telomeres, including Hutchinson-Gilford progeria syndrome. So this woman, she has extremely short telomeres, and she's very, very young. She's only, I believe, in her 20s, and so it causes rapid aging and deterioration, as well as a whole shortage of a whole series of other side effects, including sort of like a smaller than normal head, smaller than average height, things like that. So interesting sort of thing at the end of your chromosomes. Telomeres associated with aging, associated with disease. Some people are trying to use telomeres to reverse the aging clock, which I think is pretty cool. Kind of, I don't know if I'd want to live forever, but not having to suffer from some of the diseases that are associated with destruction of your telomeres sounds pretty cool. So with that, that's going to be the end of today's class on DNA. We're going to come back and talk about DNA again in the future. Again, it's a really complex sort of topic, but this is the basics of DNA. And we do this sort of thinking about chemistry and then energy and then DNA, because we need to set those three things up to understand our next topic next week, which is going to be evolution. So we're going to take a two-part look at evolution next week, starting Monday. We're going to look at first the process called natural selection, which is sort of the mechanism behind evolution. And then we're going to look at sort of this concept the following day, how we can use natural selection to make new species. And then once we sort of get that home, we're going to talk about how life came to be. We're going to talk about microbes, and then we're going to talk about all sorts of cool biology after that. But these sort of the next two classes are going to be focused on evolution. And we need to understand DNA, energy, chemistry to understand evolution first. So with that, I hope you guys enjoyed today's class. If you have any questions, please I will hang around as always. But I hope you guys all have a good weekend. Please remember that you do have an assignment due on Friday. You can, it's due tomorrow night at midnight, but there's no like time clock on it. So if you if you like say started it yesterday, it's still available for you today. There's no like you have to finish it in an hour. You can just do you can do it over the course of the week if you want it. But it is due by midnight. Anytime after midnight, remember it does become late, and then you have a week to do it. There is, I think, 9 questions on the first weekly assignment. There are 5 multiple choice and 4 short answer. The short answer ones can be kind of tricky. So I would recommend taking a peek at them. And if you don't quite get it, shoot me an email or post a question on the Blackboard discussion group. They can, I always find short answer questions to be easier, not to grade, but to answer, because, there's sort of wiggle room. And I can, if I think you're on the right track, I can give you points for that. But multiple choice, it's like yes or no. So anyways.
I noticed on one of the short answers, you said to cite where we got the answer from. Is it okay just to reference the slide, like the number of the slide in the PowerPoint?
Sure. Which are you talking about? The one that asks about trace element deficiency?
I think that's what it is. Yeah. And at the end, it just says, make sure to cite where you got your answer.
Yeah. Yeah. If you want to, I was hoping that you guys would go towards finding a new trace element deficiency, but because there's a lot of them. But if you want to talk about goiter, that's okay too. And you can cite the slides, but you're probably going to you might need a little bit more information. Well, I don't know. Yeah, but if
Okay.
you want to cite the slides, great. But if you look at something we didn't look at, like say, for instance, manganese deficiency, you know, if you go to say, you know, Johns Hopkins University, let me know that's where you got it. Just because otherwise, I don't know if you just
Okay.
copied that
Yeah,
information.
I
Yeah.
cited the slide that I got the rest of the information from, but the disease that I actually like gave an example of, I just knew about. So that's why I did that one instead of like doing further research.
Okay. That's fine.
Okay. Thank you.
Mhm. Thank you, guys. Your all mics are off, so I don't know. Usually when I get to this point, no one's around. I'm going to end the